Welcome, Neet aspirants. So, today we see a small topic from the photosynthesis where most of the students will have a clear confusion in this topic. That is chemiosmotic hypothesis. So, what is called as a chemiosmotic hypothesis? So, the chemiosmotic hypothesis explains how ATP and NADPH are generated. So these two are the products of light reaction, that is ATP and NADPH. So the products of light reaction are utilized in the dark reaction for the synthesis of the food, that is glucose. So now see how the ATP and NADPH are generated. So you know that it happens in the thylakoid. So the thylakoid membrane consists of photosystem 2 and then photosystem 1. So the photosystem 2 that is having the nanometer of 680 and photosystem 1 that will be having the nanometer of 700. So the photosystem 1, near this photosystem 2, the splitting of water molecule occurs. So that is, the splitting of water molecules generate electron and then proton and then oxygen molecule. Here the proton will get accumulated inside the lumen of thylakoid. So whereas the oxygen that diffuses out in the stroma region, and then the electron will be observed by the reaction center, that is one molecule of chlorophyll A of photosystem 2. So this you know that non-cyclic photophosphorylation, so that links with chemiosmotic hypothesis. And in photosystem 2, the electrons will get excited and it is accepted by the primary electron acceptor, that is phaeophytane. From the phaeophytane, the electron will be carried out to the electron transport system or electron transport chain. So where the electron is first observed by the plastoquinone and then it moves towards the cytochrome B6. So the cytochrome B6 that passes an electron from plastoquinone to plastocyanine. So here when the electron moves from plastoquinone to plastocyanine, so that time the proton from the stroma region will be accepted by the cytochrome B6 and the cytochrome B6 also pay one of the way for the accumulation of this proton. So in two ways the proton gets accumulated. So one is that is due to photolysis and the second reason is the cytochrome B6, it is the proton carrier. The cytochrome B6 is a proton carrier. So whenever it is passing the electron from plastoquinone to the plastocyanine, so that time it fetches one of the proton from this stroma region and deposit in the lumen of thylakoid. So because of this, the proton gradient will become high inside the lumen of thylakoid, so where it becomes low in the stroma region. So inside the lumen it will be having the high electrochemical potential gradient and outside it will be having the low electrochemical potential gradient. So inside the pH also will become high. So whereas in the stroma region, so the pH that will become low. And now the high concentration of this proton, that is proton gradient, so should come out from this lumen to the stroma region. So the membrane does not directly allow the passage of this proton. So here it should be carried out with the facilitated diffusion. So what is called as a facilitated diffusion? Directly it cannot pass through the membrane, but with the help of this, the carrier protein. But with the help of this carrier protein, the protons can come out to the stroma region. And this type of diffusion we call it as a facilitated diffusion. So here the carrier protein is divided into two, that is the CF0 particles and then CF1 particles, and the proton, it passes through the CF0 and then it passes down to the CF1. So when it passes to the CF0, the enzyme ATP synthase that is present in the F1 particle, it will get activated. So the enzyme will get activated. So when the proton reaches the CF0 itself. And then, so when the proton passes through the CF0, the enzyme ATP synthase, otherwise we call it as the ATPase, so that will get activated. So the adenosine diphosphate combines with inorganic phosphate and then it produces the ATP. So this how the ATP is generated in the photosystem 2. And then, so next we see another energy molecule NADPH, how it is formed. So when the proton from this lumen reaches the stoma region, it will go and reduce the NADP plus. And then because of this, the proton that gets joins with this NADP plus, they get reduced to NADPH. So as a result of this, the second energy molecule, that is NADPH, is formed. So chemiosmotic hypothesis explains like how ATP is synthesized. So here the proton gradient that drives the synthesis of the ATP, as well as the NADPH is formed. That is the strong energy compound, that is NADPH is formed. So one NADPH2 is that is equal to the 3 ATP molecules. And all these, that represents the chemiosmotic hypothesis, and this was first explained by the Peter Mitchell. So this person explains the chemiosmotic hypothesis. So hope you all understand what chemiosmotic hypothesis now. If any doubts, please comment in the comment section. Thank you.